amazing. Perfect. So we're looking forward to sharing our research with you and encourage presentations. We encourage questions after each presentation today. There will also be the opportunity for a short discussion period with both presenters at the very end of the session, so stick around for that. So on to the main event. I'm so pleased to introduce Dr. David Olson and Dr. Gerlinda Metz today. Dave Olson is a University of Alberta Distinguished Professor of Obstetrics and Gynecology with adjunct appointments in the departments of Pediatrics and Physiology. His work focuses on improving women's pregnancy and newborn health. He's a firm believer in the importance of translating scientific discoveries into products that serve society. Gerlinda Metz is the professor at the University of Lethbridge and an adjunct professor in the Department of Obstetrics and Gynecology at the University of Alberta. Her research program focuses on the influence of stress and other environmental factors on maternal and child health. Her groundbreaking mechanistic studies of transgenerational programming by stress are now leading to the discovery of new biomarkers for risk prediction and early diagnosis of disease. So we'll start by inviting David Olson to lead us off with the first presentation. Dave, the floor is yours.
Well, thank you very much, Galicia, for that nice introduction. And please bear with me as I try to navigate screen sharing. Does that work? Everybody see my slides? Good. Well, thanks again for that introduction. And I'm going to start us off on some studies related to prenatal maternal stress and its effects on the pregnancy outcomes.
Dave, we can see your second slide. I don't know if there's a presentation mode. Maybe you can.
I am in presentation mode.
I think you need to swap your displays really quick.
Say again?
I think you need to just click the swap displays button really quickly. It's flipped around for your screen, I think.
How's that?
Well, that didn't do anything. We're seeing your presenter view.
Should I go back to, I'll get off, I'll try, I'll stop sharing and I'll try to share again, okay?
Okay.
We did rehearse this, folks, but it didn't, I'm not sure which screen let's try this one. That better?
It's still in presenter view.
I am in presenter view.
Try use slideshow at the top, Dave, and see if that helps.
Top left.
I'm not sure where slideshow is.
In the top left of your screen, there's a swap displays and then a use slideshow button.
Oh, okay. Is that better?
Black screen for me right now.
Try to advance the slide, maybe.
You know what? Why don't I'm going to stop sharing. I'm going to get out of presentation mode and then I'll get into it. Sorry about this, folks. We did rehearse, but the rehearsal was
Dress rehearsal went too well.
yeah. Sorry, I'm just a little, I don't know what to do here.
Do you want us to share the slides, Dave?
No, I'd rather do it myself.
Okay.
Back to the meeting. Okay. Oh, here we go. Alrighty.
I think so too, Saya.
Can you see that?
Yes.
Yeah.
See my title?
Yeah.
How does that work? Can you see my title now?
Yeah, we can see the slide. We can see the notes to the side, but I think that's fine. I think we can just continue with that. We can still see your slides well.
All righty. Well, I apologize for these. Delays. I'm not very good at this. So I want to share two stories with you to begin with. And they're two different kinds of stress stories. And as you know, I study preterm birth. It's my main adverse pregnancy outcome. But I would, I also will discuss some other pregnancy outcomes today as well. So you may all remember that 6 years ago, that the Humboldt Broncos hockey team bus hit a truck in Saskatchewan, killing 16 and injuring 13. Kelsey Fiddler was stopped at the same intersection and saw the crash with her sons. After she called 911, she brought pillows and blankets to the survivors. She was 28 weeks pregnant. She started to have contractions every 15 to 20 minutes. So she drove herself to the nearest regional hospital when the contractions were 5 minutes apart. She was then taken to the tertiary center in Saskatoon. However, her contractions stopped and she delivered at term. So why, after seeing this very traumatic, stressful event, did she start to go into labor, but really didn't finish and delivered at term? Let's contrast that to studies done back in the 1950s with Dougal Baird from the University of Aberdeen. He looked at the type of social class or status that people lived in and their rates of prematurity. And he monitored the wife's social class by looking at the wife's father's social class. 1 and 2 were the highest classes, and 4 and 5 were the lowest classes. And the same for the husband's social class. 1 and 2 were the highest, and 4 and 5 were the lowest. And remember, back in the 1950s, Aberdeen was kind of a fishing town, and people lived in poverty in the lowest classes for generations. But what they learned was that the lowest social classes for both men and women had the highest rates of preterm birth. So what can we learn from this? Well, we're going to compare different kinds of stress today on adverse pregnancy and offspring outcomes. We'll look at gestation length, behavioral development, neural development, metabolic development, depression and anxiety, and I'll even share with you one qualitative analysis. I'll examine the role of resilience, the effects of environmental influences, will lead ultimately to a definition and role of allostatic load and some proposed mechanisms based on that. And then I'll finish off very briefly with some clinical needs and applications. And I would like to thank Suzanne King from McGill University for sharing some very important slides with me about her study on the January 1998 ice storm. And many of you may remember this, but in early January, the weather warmed up and then it suddenly began to rain. But as the rain occurred, then the weather and temperatures plummeted again, and all the water started to freeze on the power lines, and the power lines came down. So about 40% of the province was without power for up to 40 days. And this had affected millions of people. And she then decided that she was going to recruit a cohort of women who were pregnant during the ice storm and study them. And I'll share some of her results with you. So the first is gestational age. And if you look at the red line here, you can see that women who were in preconception and then during the first trimester and a little bit of the second trimester had shorter gestations. They went from about 40 weeks down to 38.5 weeks. And then during these 2 critical periods of gestation, and then afterwards back up again to normal. Now, this is not preterm birth. Preterm birth would be below the bottom line here, but there was a shortage of gestational length in her study. She also then queried the participants in the study about their stress. She had two different kinds of stress examinations. One was documented by taking stress questionnaires, and this would be called subjective stress. And the other was objective stress, where the women were just asked about their feelings about the stress and how it affected themselves and their lives. And so some subjects were low subjective stress, and some subjects were high subjective stress. And if you look at this graph, the x-axis over here is increasing number of days of pregnancy of the ice storm. So the women who were exposed early on in the longest period of time to the ice storm had babies who were fussy and difficult. That's the green line here. So these women with the longest exposure and the highest subjective stress had the fussiest babies, whereas the women with the least subjective stress had babies who were less fussy and difficult. Now, if we continue to look at the offspring from these studies, you can see that women who experienced high objective stress had children whose productive language was half of those with low objective stress and even lower, slightly lower levels of receptive language. And then she studied offspring and gave them the test, the AASQ test, which is an autism measure. And she found that women with the highest levels of objective stress had children who registered the highest in the autism measuring test, or risk for autism. Looking at metabolic outcomes, she found that at age 5, children whose mothers had high objective stress, that is, felt the stress of the ice storm tremendously, had a much higher rate of obesity. And this continued throughout childhood as well. And they also then later on developed signs of type 2 diabetes. And one of the examples of that is comparing maternal objective stress exposure with offspring's insulin secretion. And there was a positive correlation with that, which is a harbinger of type 2 diabetes. And these were in 13-year-old offspring. So that, I just want to go back to this and say also she, and I don't have the slide, but she found higher levels of inflammatory cytokines in the plasma of offspring, adolescent offspring of women with high objective stress. And if you go back and look at her overall studies on metabolism, children as early as 5 were showing signs of obesity and overweight. Children at age 8 were showing signs of type 2 diabetes, and children at age 11 were showing signs of high blood pressure. So clearly, maternal stress has effects on offspring, metabolic, and neurodevelopment. Then I was able to recruit her and others to study the Fort McMurray fire here in Alberta in 2016. 88,000 people were evacuated, and we figured that we had about between 1,200 and 1,500 women who were either pregnant or postpartum, maybe a few preconception in this group as well. And we looked at resilience in these women, again with objective stress. Now, this vertical green line you see in these two graphs is just to separate low stress from really high stress people. And we also did a test to look at relative resilience. And we found that women who had high resilience had fewer PTSD type symptoms, but that only lasted when the objective stress was relatively low. When the stress got higher, then the resilience was overcome by the high levels of stress. We also, a year later, were able to take an opportunity to study women in Houston, Texas, after Hurricane Harvey hit and flooded Texas, and we recruited well over 1,000 women into this study. And I just want to show here that women from the Harvey study in blue and the Fort McMurray study in red had very, very high levels of anxiety and depression and peri-traumatic distress rates in different kinds of measures that we looked at. And Suzanne King was able to study also, here's the ice storm, but also the Iowa flood study from 2007. And she was able to study the Queensland flood in Brisbane in 2011. And these two stressful events, the fire and the hurricane, were very, very stressful. And clearly these women had prenatal maternal stress that was really quite high. So these natural events, natural disaster, traumatic events can really impact women's anxiety and depression levels. And this obviously, as I've shown, has consequences. And I want to share one qualitative study with you. We did we asked the women in Fort McMurray and in Houston, we asked 25 in each group to do a expressive writing exercise. And they were to write for 15 minutes a day for 4 different days on subjects that or topics that we gave them. And I want to report here just the qualitative outcome from one. We asked 50 women to describe whether the wildfire or the hurricane was the most traumatic event they had ever experienced. And to our great surprise, only 10 out of the 50 said that it was. The other 80% said that the wildfire or the hurricane reminded them of previous traumatic experiences that they had in their lives. And these other experiences were such things, they were really quite severe. They were things like abuse or the loss of a loved one, like a child or a spouse, or other kinds of losses or issues that destabilize the relationships with important people in their lives, and or even previous motor vehicle accidents that they had. But what we learned from this is that I don't think it's so simple for us to do studies on individuals just based on one traumatic natural disaster event, because people have a number of traumatic events throughout their lives. And this traumatic event, the wildfire or the hurricane, made them remember other examples of trauma they had in their lives. And we have to factor that in when we do cohort studies. So what do we learn from these? Well, maternal depression and anxiety are high after traumatic stress. It also makes individuals recall earlier stresses. But the stress, while the stress is great, it's not great enough at this time to cause preterm birth. Several offspring health problems are obviously evident: fussiness in babies, and increased risk for neurodevelopmental disease and autism, and increased risk for metabolic disease. And we learned that high resilience can mitigate to some degree, but not all stress effects. Now I want to examine a couple of examples that look more at environmental and chronic stressors, not the one-off traumatic events. And I want to share with you a wonderful study done by Inga Christians, a graduate student in the lab, that looked at chronic life stress in women during their childhood. So with Inga and Kathy Hagedorn from the nursing faculty, we put these 8 different stress questionnaires together, and then we queried women who had just delivered either a preterm baby or a term baby. So it's a retrospective kind of examination. We had 223 questionnaires, and 2/3 were controls and 75 were preterm birth cases. And what we discovered from Inga's work is that if a woman had 2 or more high adverse childhood experiences out of a group of 10, and I'll share what they are in a minute with you, she had double the risk for having a preterm delivery. And if a woman was abused, either as an adult or a child, she had a 30% increase in risk for having a preterm delivery. Significantly, if a woman had 4 or more adverse childhood experiences, her risk was 50% for having a preterm delivery. And this is a list of what the adverse childhood experience questionnaire is. There are 3 questions related to childhood abuse up to the age of 18, a neglect and dysfunctional household. So here we have more chronic kinds of stresses that women experience, and these seem to be associated with preterm delivery. We then did a systematic review of the literature looking at childhood abuse and intimate partner violence. And we looked at 8,000 different papers, and we narrowed it down to 16 that fit all our criteria. But what we learned was that in the literature that childhood abuse is associated with having a preterm delivery. And recent abuse as well is associated with having a preterm delivery. And we also learned that this is associated with having low birth weight babies. We were then able to take the same data from Inga's study, and with the wonderful assistance of Matthew Guzdial of our computing science department and his student Samriti Vaid, they were able to use artificial intelligence and machine learning methods to determine whether or not we could get a predictive algorithm out of this. So they used deep neural network approaches with transfer learning techniques, which are very important for small number data sets like we had. And they were able to actually predict every one of the preterm deliveries. The preterm deliveries are indicated in this graph with the solid black dots. That was the actual date at which the preterm deliveries occurred. The predicted date were the open circles. So the blue lines are predicted early and the pink lines are predicted late. But overall, their variance or standard deviation was only 6 days of the mean value. So this is a very good predictive tool. Not only does it predict the risk for preterm delivery, but it predicts when. And of course, it's almost unbelievable. So we need to test it again in more retrospective studies and then do a prospective study. But this is the direction that some of this work can go. And I'm sorry, we used, they used the data from the questionnaires to create an allostatic load or a stress index that was fed into the algorithm. So summing up, stresses in preterm birth, well, pregnant women and all of us are exposed daily to tolerable stresses. And we have pretty good coping mechanisms to deal with these. And these lead to normal health outcomes and for pregnant women, normal pregnancies. But then when intolerable stress comes along, either a sudden one or a chronic one, or it could even be an inherited stress from mothers or grandparents, or if there's a secondary insult, such as inflammation, that's added on top of the transgenerational stress, then adverse health outcomes can occur through epigenetic and inflammatory changes, and this can lead to preterm birth or adverse pregnancy outcomes. And Lazarus and Cohen began to model this stress and disease many years ago, where they said that the environmental demands, when they exceed the adaptive capacity of an organism, this can result in psychological and biological changes that may place persons at risk of disease. Then Bruce McEwen from Rockefeller University coined the term of allostasis. Now, allostasis differs from homeostasis because you do not return to a set point. But allostasis is sort of the accumulation of lifelong stress. And when we're in a state of chronic or repetitive stress, we get to a prolonged allostatic state or a new set point that we don't return to the original set point, but we have to learn how to adjust to a new set point. And this is called, the total burden of stress is called allostatic load, the cumulative results of an allostatic state that wear and tear on the body and the brain. Suzette Bramall-Phillips and I tried to put this together into this diagram. Along the x-axis is time and the accumulation of stressors. And increasing allostatic load is going down on this figure, as you can see on the right side y-axis. And the healthy state or well-being and resilience is going up on the left-hand side of the y-axis. So if you look at the blue line, as an individual accumulates increasing levels of stress, it can be transgenerational, lifelong, and so on, then their allostatic load increases. And it gets to a certain point where they get to their resilience threshold. And if they exceed their resilience threshold, then they're at risk for an adverse outcome, as you see in the bottom half of this chart. We are hopeful, and we have evidence, and Dr. Metz will present some of this evidence to you, that positive interventions, though, can mitigate the level of allostatic load, or what they can do is change the threshold so it takes a higher level of allostatic load to get to a danger point in terms of stress. And so we hope that going forward that positive interventions can reduce the adverse effects of too much stress. More mechanistically, an individual inherits some generational experience, and then they experience their own life adversity and stresses, and they develop their own responses to this. If their allostatic load, however, exceeds a level that's too high, then this can lead to an adverse pregnancy outcome, such as preterm birth or some of the others. And not only that, but they have the danger of passing it on to the next generation. And for those who are placental biologists in the group, even more mechanistically, this maternal stress we know can lead to increases in corticotropin releasing hormone in the placental tissues. We know it leads to more cytokines, more prostaglandins, and of course, higher levels of maternal cortisol. The danger in this is that cytokines from our work can inhibit 11-beta-HSD2, that is the enzyme that converts cortisol to the more benign cortisone. And they also will stimulate 11-beta-HSD1 that converts more cortisone to cortisol. And the consequence is that there's more maternal cortisol, and it will cross the placenta into the fetus, along with more cytokines that can cross over to the fetus, stimulating more prostaglandins locally. And these combinations together can lead to many adverse neurodevelopmental and behavioral and health problems in the offspring. I want to share one other study that was done by a resident in our obstetrics department, Alicia Long and Atusa Gulfer. And this has to do with what now can we do about this in the clinical situation, and what can practitioners do in terms of altering their practice. So we asked the question, what is the practice in Edmonton? And so these ladies queried 58 Edmonton obstetricians and gynecologists, and they got results back from 49 of them. And what they learned was that a third never or rarely screen for intimate partner violence, one of the factors that we identified as being a risk factor for preterm delivery. 69% never or rarely screen for childhood abuse, which we showed has a correlation with the preterm birth. Nearly all of them do not have any screening protocol, at all, and 3/4 of them do not have written materials to share with their patients or any other kind of advice or materials to share with their patients who might have had an abusive situation. And nearly all feel that their screening is inadequate. And in part, this is due to many barriers that exist in the prenatal assessment situations when women go in for the prenatal visits. So clearly we need to learn how to screen better, and we need to provide practitioners with tools to help their patients if they find someone who has had an abusive situation. So summary, we think our data suggests that chronic, lifelong, and generational stress, like allostatic load, includes all of the adverse outcomes of traumatic stress plus preterm birth. And chronic and traumatic stress are really just different parts of the same continuum. So the examples I gave you at the beginning were different parts of the same continuum. High allostatic load may be predictive for preterm birth risk or other adverse health outcomes. And stress mechanisms during pregnancy are still being defined, and we still have more work to do on that. And we need better translation of our evidence into clinical practice to avert more adverse pregnancy outcomes. So we need better understanding of father's stress as well, which I didn't mention today. We need better interventions to mitigate the adverse stress effects, and we need better translation to public health and healthcare. And I'd like to just finish by thanking my wonderful collaborators here at the University of Alberta. And this is our 20th anniversary year of collaborating with Gerlinda, and I'm very thankful for those wonderful 20 years. And Suzanne King has been a wonderful collaborator, and Joanna Bick down in Houston as well, and our funders, the CIHR and WCHRI. So thank you very much, and I'll take any questions you might have.
Thank you so much, Dave, for that excellent presentation. I'm going to open it up to the audience to ask questions. Please use the raise hand feature to ask your question. I'll do my best to invite you to voice your question in about the same order that hands are raised. And if you're more comfortable using the chat feature to ask your question instead, we will do the best that we can to voice your question for you. If we get the substance of that question wrong, please just unmute and help us get the question right. We're really looking forward to all of your questions, so please don't be shy. I can start us off with a question for you, Dave. I was curious, a lot of these studies that you were presenting today, they're presenting objective levels of stress, like self-reported levels of stress. And so it seems that how individuals perceive their own stress and the impact of that is even more impactful, I suppose, than the actual events of stress that happened. Can you talk a little bit about that and like what that means for coping mechanisms and resilience building?
Probably not. That's really beyond what I know, but I certainly think that individuals are aware when they're in a stressful situation, and are probably, I think there's a lot of information, in the press and every day, and I have a suspicion that people are particularly sensitive to that. And even after COVID, individuals, and I know myself, we expressed stress behavior. So I have a feeling that people are more cognizant of it, and I think they're more anxious to find good solutions to the problem as well. I can't tell you, though, about how well individuals perceive their own stress. I just think that people are more aware of it, and and it could be measured through the stress questionnaires or through self-examination.
Mm-hmm. Marilyn, would you like to ask your question?
Sure. Hi, Dave. Thank you so much for a really interesting presentation. So for the data that you presented on obstetricians and gynecologists who are facing barriers in terms of the screening protocols that they were able to use in their practice, I'm curious what specific types of barriers that they mentioned facing. if you could talk a little bit more about that.
Well, I think one barrier would, I mean, they're cultural, certainly, also time. You know, how long is the average prenatal visit? 10 minutes? And in Alberta, we have a form that is filled out, and way at the bottom of the form is, have you experienced any interpersonal violence? And oftentimes the obstetrician never gets a chance to get to the bottom of the form and ask those important questions. So that's part of it. I think another one is that they don't know how to ask the question. I think that's something that came out of the study. They're not sure of how to ask the question, and they're not sure how to respond if they were to get a positive answer as well. So, and then, like I said, cultural and other kinds of background kinds of considerations are in there as well. I know that in some countries, the spouse, the partner will accompany the woman into the prenatal visit, and then it becomes very awkward to ask if you're experienced any abuse or violence in your life. So that's another barrier in some jurisdictions that gets in the way.
All right, thank you very much.
All right, Zubia, would you like to ask your question?
Yeah, sure. Hi, David. My name is Zubia Mumtaz, and I'm in the School of Public Health. I'm not sure if mine is a question, but we are just in the tail end of collecting data to understand some of the social risk factors of preterm births in immigrant women. And our early analysis is starting to show that, yeah, it probably is stress that is related to the various dimensions of immigration. Some of them have issues regarding, the status in the country. Others have issues related to getting their jobs, and they're underemployed or unemployed. And we didn't come across any interpersonal violence. And that probably is because of, I don't think people are willing to share that piece of information at the first meeting or even a second meeting. But yeah, it's very interesting to know that, and these findings were coming out of our qualitative data. So when I saw this presentation coming up, I was very keen to attend it. And it's wonderful to see all the mechanisms where you have identified the potential routes, and that some of the routes for immigrant women may be completely different.
Absolutely. I think Dr. Metz is going to share an interesting slide with you, some data from Pierre Buchan's. And we can talk about that later if you'd like as well, regarding immigrant women and their daughters. Thank you for your comment. That was very important.
Yeah. I'll reach out to you once our data analysis starts.
Okay.
Thank you.
Thank you.
We have a question that came through in the chat. Carla says, hi, great presentation, Dave. You mentioned that the stresses that pregnant women experience has an effect that may persist across generations. Can you discuss a little bit more how that is seen in the affected generation and if there are current actions to mitigate that?
Well, it certainly exists, Carla, and I'm going to refer you to Dr. Metz's presentation because she's going to discuss this in greater detail, including interventions. So just stay tuned. I think you're in for quite a ride.
Perfect. And there's one more question that just came up in the chat. The offspring born during the ice storm may now be having their own children. Are you looking at the outcomes of the next generation as well?
Well, I wouldn't be, and Dr. King has now retired, so I don't, maybe somebody else will pick this up. Marilyn, you're at McGill, maybe you know somebody who might pick it up. All I can tell you is that back in 2018, when these offspring were 20 years old, there was a news story done in Montreal and probably on Global and throughout, and then I saw it here in Edmonton. But the 20-year-old offspring were trying to get a job working in Dr. King's research group. And one young lady said, I'm I'm very anxious, and I understand why I'm anxious, and I want to learn more about why I became this way and what I can do about it. So I think that that generation is aware, and they're anxious to get involved in learning some answers.
One more question in the chat from Anna McKinnon and her lab. Thanks for sharing this great work. For the results that show higher risk for child outcomes like ASD, how do we avoid fear mongering among pregnant women who are already worried about impacting their baby?
Well, it's a risk. That's a good question. And I would need to, again, talk with Suzanne King about this, but the risk is increased. The actual numbers of children with autism may not be as high. So that might be a mitigating, at least a reassuring kind of response. I don't know how to overcome fear mongering. I think we have to be honest about what the test results are and then prepare for them. But I think there are better therapies and interventions these days than there used to be as well for kids with autism.
Perfect, thank you. So that's about all the time we have for now, but don't worry, there will be time for additional questions at the end. So if you have any more you're thinking of, save them. And next, I will invite Gerlinda Metz to the virtual stage to share her important work with us.
Thank you very much, Kalisha, for your great introduction. And thank you to WCHRI and Dave and Michelle and Carrie and the organizing team, and also John for hosting the session today and for inviting us to be here. It's really a pleasure to follow up with some of Dave's excellent thoughts and ideas. Thank you for going ahead, because I have big shoes to fill here. But bear with me, I'm really happy to contribute to the discussion today about potential mechanisms of some of the phenomena that Dr. Olson has been talking about. So, as Dave already mentioned, stress during pregnancy is something that is not very rare, unfortunately. About 80% of pregnant mothers would usually report that they experienced mild to moderate or even some severe distress at some point during their pregnancy. And recent studies, including some from Dr. Giesbrecht's lab and many others in Canada and around the world, have reported that even during the COVID pandemic recently, some of these levels of stress went up further. And along with that, there were also generally reported higher rates of anxiety and depression among pregnant women. Of course, we need to do more mechanistic studies and understand these underlying biological pathways that contribute to these observations. So, as Dr. Olson already mentioned, stress and adverse mental health outcomes are very closely connected, and taking into account also the effects of the recent COVID pandemic, it's important to consider the effects of social isolation and perceived loneliness as well. So all of these influences can potentially affect the maternal health and pregnancy outcomes, and ultimately also show trickle-down effects to the developing fetus, especially epigenetic programming that ultimately will affect gene expression patterns, which downstream can alter cellular metabolism and cellular function to affect lifetime health trajectories of the child from early development to old age. And we're trying to put this together in animal models, so it helps to discover some of the mechanisms using rat and mouse models. So here I'll be talking about a rat model, and this work is a result of the 20 years of collaboration like Dr. Olson already mentioned. We have a kind of a anniversary going on here this year, and it's really a pleasure that we had the opportunity to start this kind of research in 2004. So Dr. Olson came down to visit my lab at the University of Lethbridge, and we sat down over a cup of tea. And he asked me, how would you think about preterm birth? What would cause preterm birth? And how can we develop a better animal model of preterm birth? Because at that time, there were only pharmacological means that would induce preterm birth. And so because I was interested in stress as a neuroscientist, I spontaneously said, of course, we'll try it out. We'll use a stress model. And we started with a mild to moderate psychomotor stress model of restraint and swim stress in a group of 5 dams. 2 of them were controls, and 3 of them were stressed, and 2 of them delivered early. So then we wrote grant applications, we got a little bit of funding and did a more controlled, larger study. And what you see is the outcome here, that actually the parental generation we're talking about here is filial 0 generation. The non-stress and stress groups were not significantly different from each other in terms of gestational length. But I'll come back to this graph, so bear with me. It just so happened then that we got back together and we sat down again and thought of, why would these results be any different from our pilot study that we did before? And Dr. Olson came across a presentation that he listened to by Dr. Pierre Buchans from Tulane University. And here Dr. Buchans was talking about birth outcomes and pregnancy experiences in migrant women, including women from Algeria and from Mexico. And it seems they had healthy, normal pregnancies, but their daughters were at higher risk of preterm birth. So this gave us some further ideas to dig a little bit deeper. But before I'm moving on, I would like to talk a little bit more about these epigenetic markers. We hear a lot about epigenetics. That means these are markers that are being generated independently of DNA sequence, but they can also affect gene expression patterns. Some of these markers, for example, microRNAs, these are post-transcriptional silencers of gene expression patterns. So the higher the microRNA level, the lower the gene expression product. And Dr. Fabiola Zucchi, former postdoc in my lab, has investigated microRNA expression in pregnant dams exposed to no stress conditions or mild to moderate experimental stress. And you see that in the black bars. Some of these critical microRNAs that were actually involved for maternal health, depression, and anxiety-like symptoms in our rats were downregulated by gestational stress. So this gave us something to think about. How could we model these findings? And we investigated then how these epigenetic signatures of stress can be passed on from one generation to the next. This research, unfortunately, is quite impossible in human populations. So we do have to rely on animal models here that have a shorter lifespan. And originally, we would assume that these epigenetic markers being passed on help prepare the offspring for better survival and adaptation. So the mom is passing on the message to her baby that it's a stressful environment out there, and the child has to be prepared metabolically for a stressful environment, also behaviorally with adapting to a potentially dangerous environment after birth. So it's a very potentially adaptive and helpful mechanism that we're talking about here. So then, instead of just looking at daughters, we went a little bit beyond this model and looked at 5 generations of pregnant rats in our lab. This is a logistic nightmare to work with time pregnant rats across 5 generations. But we succeeded, and we generated at least 3 different lineages in our lab continuously. I'm talking about an unstressed lineage where there was no experimental stress in any of the generations of rats. Then I'm also referring to a transgenerational stress lineage where stress was given only in the parental F0 generation. And then the pregnant daughters, granddaughters, and great-granddaughters did not receive any further stress. We also included a multi-generational stress lineage where every time the daughter granddaughter, great-granddaughter got pregnant, we would stress them again so that we had a multi-generational stress model where we would hopefully see some adaptation over generations and hopefully also the development of resilience. So I'll show just a few little pieces of these data that we gained from many iterations of these studies over the years. So here I'm going back to my original graph that I showed with F0 non-stressed versus F0 stressed animals being compared. That did not show a shortened gestational length induced by stress, but look at the daughters, F1, and the granddaughters, F2 generations, where you actually see now a significant shortened gestational length. And the gestational length was shortened up to about 13 hours on average. If you go any shorter, you actually end up in rats with non-viable offspring. So this showed us that actually there is a transgenerational or multi-generational stress effect on gestational length, raising the risk of preterm birth. This was the first animal model where someone was able to show spontaneous preterm birth in a controlled environment in the lab in rodent models. And we were then digging a little bit further because I mentioned the epigenetic markers that are very important. And we did look at many different epigenetic mechanisms here, including microRNA expression, these post-transcriptional silencers of gene expression. And we did find in the F1 and F2 generations, there was, in addition to the shortened gestational length, an increased level of microRNA 200B. MIR200B by Coral Mendelssohn's lab in human studies, human tissue studies at Houston, has actually shown that MIR200B expression is being altered in the link to preterm birth, and it's now actually serving as a biomarker of preterm birth. So we did see this altered in a very reliable fashion across generations. We also looked further, and of course, we were interested in changes in gene expression patterns downstream. And here you see mRNA expression levels in male and female fetuses and their cortex and placenta. And we've seen that actually there is a difference in the stress lineages compared to control non-stress lineages. That is in a very robust fashion. And we have only very few reliable pathways that are being altered in a reproducible fashion by trans and intergenerational stress. These effects also then have downstream effects on metabolism in the cells. And here's some data by our group in collaboration with Tony Montina, our NMR facility manager and director, who has helped us study metabolomic profiles of transgenerationally and multi-generationally stressed lineages. You see those ellipses here in green and blue, and you see how they are set apart from the metabolic profiles of control animals. So we end up with having a very distinct metabolic profile that you can actually measure in solid tissues, in blood, but even in urine. And this data that you see here is from urine, and the pathways that are involved and are distinctly altered by transgenerational stress involve protein synthesis and antioxidant pathways, but also catecholamine biosynthesis, which you see here as the fourth most significantly altered pathway, which includes important mediators of the stress response and also transmitters that are involved in mental health outcomes. Now, looking at this to summarize our findings, we see that the brain acts as the master gland in triggering a stress response, which then leads to downstream epigenetic modulation of gene expression patterns, which then alters physiology and metabolic functions within the cells, ultimately changing organ function and affecting health trajectories, including those trajectories for liver and kidney and heart, but also for the uterus and preterm birth risk, and of course also for mental health trajectories and development and aging for the brain. So this made us wonder, how can we adjust our animal model better to be more ecologically valid? Because we have been realizing that social isolation and perceived loneliness are also important triggers of stress for our pregnant moms. And it so happened that just before the COVID pandemic, actually quite a few years before that, we already got interested in social isolation effects. And we did have an animal model ready, and we're breeding several generations of socially isolated rats. At least during pregnancy, we gave them social isolation. And it turned out that this was indeed a quite stressful experience. So the data you see here is a collaboration again, where Dave Olson was assisting us with interpreting the findings of gestational length changes, and Aaron Falkenberg in my lab read several generations of rats. So I'm showing you here the control group housing versus rats that were during pregnancy exposed to social isolation. And you see that here, in this case, social isolation did shorten gestational length. And it made us curious about subsequent generations. And here, we did not see a further shortening of gestational length across generations. F1, F2, F3 generations, when they were pregnant, they had normal pregnancy lengths. So this made us wonder what happened here. Maybe can we learn something about maternal resilience? And we collected a lot of tissues, including uterine tissues, and we sent them to Dr. Olson's lab, where Nayara Lopez took them on and did uterine mRNA expression analysis. And she looked at various factors, including the corticotropin-releasing hormone receptor expression, and also the gene expression for HSD11 beta 2, that is the hydroxysteroid dehydrogenase expression that Dr. Olson mentioned earlier already, that oxidizes active cortisol into inactive cortisone. And in both cases, we saw significant changes between the F0 and the F1 generations in the uterus. The F0 generations that showed preterm birth had altered expression in these two genes, but the F1 generation actually showed changes in terms of going back to control levels in CRH receptor 1 expression, or even going higher beyond control levels in the F1 generation in terms of the enzyme HSD11 beta 2. So these indicate that there are some compensatory mechanisms at work in the uterus that would potentially prevent early parturition. And this gives us hope that potentially we can learn about individual markers of resilience and how can we boost mechanisms that prevent a transgenerational transmission of preterm birth risk. Now, putting this together, we see that the brain really drives the stress response, which affects pregnancy outcomes in the uterus and child development. And overall, we see that there's a unifying pathway which is involved in preterm birth, but even some of these pro-inflammatory markers that we see altered in preterm birth are also involved in, for example, mental health issues such as depression, which is very curious. So we see that the hypothalamic pituitary adrenal axis signaling is driving epigenetic markers to differentially lead to expression of interleukins, cytokines of various kinds that create a pro-inflammatory environment. We see cellular metabolism altered that ultimately are involved in adverse health outcomes, including preterm birth and higher risk of mental health issues for our pregnant moms. So the pro-inflammatory environment seems to be important both for mental health complications and for preterm birth. And what we want to do with the New Frontiers framework that Dr. Olson talked about is to put these pathways together and see if we can find better predictors and interventions to prevent adverse health outcomes. And this is close to our hearts because we don't want to blame moms or dad for their past experiences, or we can't change our ancestor and their stresses. So we need to learn about mechanisms in order to interact with these and prevent, hopefully, the unbroken cycle of generational transmission. So if you're asking for my opinion, I think one therapy that is very reasonable is social support, and social support has been shown to build maternal resilience. I would like to show you a little bit of encouraging data here. So in terms of social support, we are seeing a causal role for oxytocin. And this is some work by Dr. Jamshid Faraji, a research associate in my lab, who has been for many years now studying the effects of social isolation and social support on the brain and on overall health outcomes. What is really interesting is that you see plasma oxytocin levels go up in females that receive social support. You see that in the left-hand graph. A little bit more than in males, actually. So females are very susceptible. Oxytocin is a peptide hormone, which we also call the bonding hormone or love hormone. It's kind of the physiological counterpart to the stress response, like a candlelight dinner equivalent to the stress response. And it will actually boost resilience and better stress coping. We also see, and that's what Jamshid has shown, that there are even genetic factors and telomere length affected by social support. So here on the right-hand graph, you see telomere length, which is a protective ending on the chromosomes, and a longer telomere would indicate a younger biological age. So telomere length is a very robust marker of biological aging. The longer the telomeres, the better the health outcomes, the younger the tissue would be. And you see that especially in the females that received social enrichment and support, the telomeres tended to be longer. This is very encouraging news. And by further digging deeper into the data, Jamshid found that higher oxytocin levels are linked causally to higher brain-derived neurotrophic factor expression. And BDNF is the most abundant neurotransmitter in the brain, but it also is being found in the autonomous nervous system. It also can be found in organs, and it is being expressed by immune cells such as T cells and macrophages. So higher levels of BDNF are linked to more resilience, but also lower pro-inflammatory markers. And some recent work by Dr. Keiko McCreary, a graduate student in my lab, has shown that higher social support indeed drove down the expression of pro-inflammatory cytokines. So we do have a really nice pathway that we can use to generate insights into mechanisms, but also gain diagnostic and predictive biomarkers. Now, this really raises the question, how can we better translate these findings from our animal models back to human populations where we do have urgent needs to address health issues such as preterm birth? So we had the pleasure of working with a fantastic team of collaborators, including Dr. Suzanne King, who's directing the 1998 Project Ice Storm study, and the 2011 Queensland flood. We also had the pleasure of working with other natural disaster situations affecting families, including the 2013 Alberta flood. We also worked with individuals who were exposed to trauma coming from residential school, or refugees who were exposed in the past to violence and war experiences. We've also worked with individuals who experienced extreme levels of stress, such as astronauts, athletes, and first responders. But I would like to now point out some findings specifically from the Pforzheim study. So Pforzheim is a city in the southern part of Germany, and it has a high risk of non-communicable diseases because it had a very traumatic past based on World War II Allied bombings that led to 80% of the city being destroyed, and the largest rate of casualties in Germany at that time. A third of the population lost their lives. On top of this, we have current stressors such as high childhood poverty, and we also have the second largest rate of an immigrant population in the city of Pforzheim. So we do have a background of traumatic stresses of various kinds that lead to enhanced vulnerability in the population of the city of Pforzheim. So I happen to have this great pleasure of working with my colleague Rupert Linder, who is running a private practice for gynecology, psychosomatics, and psychotherapy in the city of Pforzheim. So he's on the ground there working with pregnant women who come to see him in his practice. And for about 40 years, he has used a very unique approach by using integrated personalized psychotherapy that is based on psychosomatic and solution-focused therapy, salutogenesis, couple therapy, and he uses a very specific focus on lifetime and inter- and transgenerational stresses in his approach that he's targeting with his psychotherapy. He's looking at mode of birth and gestational duration and newborn outcomes such as weight and head circumferences, and we got together to put together the data of the past 20 years of practice and work that Dr. Linder has been generated. So the results from his work are shown here. You see in orange the results of psychotherapy treated women in Dr. Linder's practice compared to, in the blue bar, standard care in Pforzheim, where women did not receive the psychotherapy intervention. You see that the rates of spontaneous birth in Dr. Linder's practice are higher, and that is due to a lower rate of C-section and instrumental delivery in his practice. On top of that, he has actually observed much lower rates of preterm birth in his population of women that he treated with psychotherapy. In blue, you see the city of Pforzheim standard care group. In turquoise, you see standard care results in Baden-Württemberg, the state where Pforzheim is located, and orange is the psychotherapy group. And you see that compared to the other rates of preterm birth, which average to about 9%, the rates of preterm birth in the psychotherapy group in Dr. Linder's practice were only 2.2%. So this is a very stunning success of an effective intervention that successfully reduces the risk of preterm birth based on psychosocial interventions. So obviously, psychosocial intervention is helping to build resilience and downregulate the effects of stress and potentially break the cycle of trans and intergenerational transmission of preterm birth risk. So I would like to summarize a little bit of what we've been discussing here and give you some further food for thought. We see that maternal stress and adverse mental health outcomes are both linked to exacerbated pro-inflammatory processes, and these stresses cumulatively program adverse pregnancy outcomes, including preterm birth, in our red model. We also see that psychosocial interventions are very effective in mitigating the risk of stress-induced adverse pregnancy outcomes, such as preterm birth. And this would give us some leads for the future to enhance stress resilience. And we see stress resilience really at the core of improving maternal mental health, but also pregnancy and birth outcomes. And so this is our idea that we're proposing to you today for further stimulating thoughts and discussion. And I would like to, before I conclude, thank my most wonderful group of trainees and collaborators who have been really central in generating these data. And it's so much fun to work together with this inspiring interdisciplinary research environment. Especially, I would like to thank Dr. Olson and Dr. Linder for their collaboration and friendship throughout these years in really helping us put these red data into clinical realities as well. And thank you to the funders. And finally, I would like to conclude with a statement by Maya Angelou, an American activist, who said, "I come as one, but stand as 10,000. With that being said, thank you everyone for being here today, and we would be happy to entertain any questions.
Thank you so much, Gerlinda. That was so interesting. I'm going to open up the floor to the audience for questions. I see Michelle. Oh, Michelle's clapping. I thought her hand was up. Does anyone want to start us off? Deborah, go ahead.
Thanks very much. It's been wonderful hearing from both of you. I've known you for a long time, and it's really great to see the work you've done. I'm working with a grad student right now who's studying the relationship between preterm birth and different kinds of maternity care practices. There have been a number of studies, including systematic reviews, that have looked at midwifery, reduced association with preterm birth, controlling for high-risk pregnancy and those kinds of things. And so, Gerlinda, what you're talking about really kind of resonates with me, and thinking about now she's trying to understand why, and she's got a bunch of qualitative focus groups with physicians and patients and all kinds of things. But what you've presented really helps me think about that potential mechanism because of the different ways that midwives practice. And so we're going to have results for Alberta, but it's going to be really interesting. I'm going to direct her maybe to connect with you and some of the work that you've done, if that would be okay.
Thank you so much, Deborah. This is great to hear. And it's so important to us to see that the data are going to leave the ivory tower of an academic lab to really reach clinical realities. And it's so rewarding to see that there are so many initiatives now being put together that are very helpful for women and families in general, and I think for a population at large and future generations. So I'm very happy to collaborate if there's any way how you see we can contribute and work together.
Yeah, I think that would be great. Thank you. Yeah, thank you both.
Thank you so much, Deborah.
Gerlinda, a change in preterm birth rates from 8 to 9% down to 2 is amazing, like more than you could ever imagine. Can you speak a little bit more to it as to the psychotherapy and what was involved in the psychotherapy in that study?
Yeah, thank you, Kalisha. This is a great point. So this is a very comprehensive psychotherapy approach that Dr. Linda is using, and he's tackling many different aspects of maternal mental health and well-being. And he even involves the fathers. He invites them to attend. They can also bring other children. It's like a very socially interactive event when a woman comes to the practice. But also I've seen how Dr. Linder is interacting with the individuals who come see his practice. An appointment doesn't take 10 minutes here, as we've already talked about before. It does take usually an hour, and it is a very subtle approach. I don't think that the women actually really notice that they're in a psychotherapy approach. It is more of a conversational style, and I've attended those sessions with women or with women and their partners. And it's a very productive environment that really transpires in terms of social support and listening and being there. And so I would call this also not only a psychotherapeutic approach, but it is a social environment where women feel safe and they feel protected, and they can talk about their past experiences and their mother's own birth experiences, which often seem to be reproduced subconsciously, unconsciously. And it seems to be a very holistic approach. And now because we have collected biological samples, we're really curious to look into biological mechanisms here. So stay tuned for a few years, and we hopefully have further news down the road.
That'll be great to see. Is telomere length something that you are considering measuring in these biological samples?
It would be something that I would love to do, but because it is still a genetic marker, it might be a sensitive issue for some of the mothers. So what we're starting with now is mainly to look at physiological markers and metabolites. And the metabolomics approach we have used is really robust in showing us which pathways are altered, because we do have the experiences from our mechanistic studies. We know exactly what we're expecting, which markers are being altered by social isolation, by stress, by traumatic stress, long-term and cumulative allostatic load. And so we can now transfer this knowledge to the samples we get from the Pforzheim study. And I think it will be rewarding to start with this angle and then follow up potentially in the future with many more levels of studies.
Definitely. Wow. We have a question that just came through in the chat for you, Gerlinda. They say, thank you so much for the presentation. I'm so excited by this research. Just wondering if there was any proposed reason for why adverse pregnancy outcomes were found primarily in the daughters, as you mentioned.
That's a really interesting question. And so what we see when I mentioned Dr. Buchan's work, it's probably in terms of immigrants, maybe the immigrants who decide to move to a new country are already quite resilient because they're taking their future into their own hands and they're moving to a different place, which takes a lot of bravery to do that. And so these women that come to another country are probably relatively healthy, I would assume, just, in general. But also, I think we see differences between social isolation versus other types of stresses. So we do have a stress component that's very specifically targeting our social isolation experience. So we do have different outcomes there. In terms of outcomes for the daughters in our transgenerational psychomotor stress lineage, I think what we're seeing is really a causal pathway involving epigenetic programming. So these markers are being found in the mother, but they're not affecting directly their birth outcomes yet. They're inherited by the daughters and granddaughters. That's per definition, epigenetic markers are heritable. And here they're starting to act preconception already, so that there's a greater vulnerability to start with. And I think this is really a key mechanism.
Thank you. So for the last, we have 10 minutes left in this session. And so to close off the session, I'd like to invite David Olson back into this conversation and just to open a discussion period for both of our presenters to answer any questions or discussion topics that anyone in the audience has. I can ask something to kick us off here. So the world is quickly changing over the last 5 years with rising levels of stress overall from pandemics, from eco-anxiety, with climate change, with increasing political unrest. And with your findings on the impacts of stress on health and pregnancy, what does this mean for society as a whole, and what should people keep in mind to focus on their health, especially during pregnancy?
Well, we've been trying to test various interventions. We haven't gotten very far yet, but we'd like to. But there are a number of mindfulness exercises that individuals can do, good sleep, exercise, good family and social relationships, and just smart living. I think those are probably going to be the best things that individuals and small groups of people can do.
Do you have anything to add to that, Gerlinda?
Yeah, I totally agree with Dave. These are important factors. There's means by psychological resilience forming ways that we can use, like mindfulness and so on. Also, it's important to keep a sense of belonging and to stay with the community. I think these are essential parts to our society, that we have a sense where we belong and what community we belong to. It's important to go out. The reason why we started to worry more about social isolation before the pandemic was actually that we saw that social media are catching up with our lifestyle, and that more people tend to spend more time at home, and a lot of children are tending to spend more time in front of a screen instead of playing and interacting in the real world. And I think it's very important that we have these personal connections in real life, and that we take advantage of the community out there to have social activities and get together and have meaningful, positive relationships with our surrounding.
There's a anecdotal observation from the pandemic. During the first 3 months of the pandemic, there were announcements from neonatal intensive care units all around the world, including Calgary, that the census, or numbers of babies in the units, was dropping off dramatically. But then as the pandemic progressed and went on, the census went back up again to where it was before. And the takeaway message that a lot of people agreed on was that at first, women in their third trimester had to stay at home, and they had to put their feet up, not go to work. And they started to relax. And so they had fewer preterm deliveries. But then with time and isolation, the isolation stress began to wear on people. And the preterm birth rates came back again.
Wow. I think that's a nice note to end on, actually, talking a little bit more about community and belonging. And speaking of community, I'd like to talk a little bit more about this research cluster, Realizing Healthier Pregnancies. But first, I'd just like to sincerely thank David Olson and Gerlinda Metz for sharing their work with us today. And thank you to everyone who joined us. The WCHRI Realizing Healthier Pregnancies research cluster is very interested in your pregnancy research. So if you'd like to learn more about this research cluster, please contact wcgrants@ualberta.ca, or stay in the Zoom space for a bit right now to talk to Michelle about participating in the research cluster. We are very interested in getting more trainees involved and involving trainee presentations into these cluster presentations as well. And if you're already a part of the research cluster, also stick around if you'd like to talk more about application opportunities or those presentations. And I'm seeing one last question actually came in, so maybe we'll just sneak that one in under the wire. So Anna McKinnon and her lab says, what about help addressing systemic barriers to accessing care and supports?
Absolutely. I think the a little study that was done by our obstetric residents highlighted that. Accessing care is a big problem. Around the world, there aren't enough mental health practitioners, and the training has to improve as well. So access to care, treatments, and all this has to be improved. So you're right on that.
All right, perfect. Well, thank you all so much for attending and participating. And please stick around and talk to Michelle if you'd like to learn more about the cluster. Thank you.